In this section, you guys are going to be reading the introduction section of your Trammell et al. Paper. And so the introduction section is going to give you a background information of why they're doing what they did in the study, as well as define some key concepts and kind of set the premises for why they did the study and what they were hoping to find. So in this video, I'm going to give you guys some background information of some terms that you might come across in that introduction section that might be unfamiliar to you. So the first one is the difference between iteroparity and simulparity. That's a really key concept to this paper. So Pacific salmon, Chinook, chum, coho, they are all simulparous. That means that they spawn once and then they die. They always die after they spawn. Pacific trout, which are steelhead and cutthroat, are iteroparous. This means that they can spawn more than one time. Now, it's not just salmon and trout that are simulparous and iteroparous. Other species of fish can be simulparous and other species of fish can be iteroparous. But for our purposes, we're mainly focused on salmon and trout here. So that's really the defining difference between what a salmon is and what a trout is simulparous versus iteroparous. So Pacific trout, including steelhead, they can spawn multiple times because they're iteroparous, but they often do not. They have to make really long migrations back to their spawning grounds. A lot of them only end up spawning once. However, a small fraction of them do end up spawning multiple times, and those are termed kelts. So a steelhead that spawns more than once is called a kelt, and that's what this paper is focused on, is what kind of factors can influence steelhead repeat spawning, and can we influence that ourselves? Another term that comes up a lot is anadromous. So anadromous is a characteristics where a fish is born in a freshwater environment. They migrate out to the ocean to mature and grow into adults. And then as an adult, they migrate back to their freshwater environment to lay their eggs. So salmon and steelhead are anadromous, which means that here it shows that they spend their freshwater phase egg through parr, and then smolts when they're going out to the ocean, and then they're living their life as adults in the ocean. Now, steelhead by definition are anadromous. However, rainbow trout are the same species as steelhead. They're exactly the same, except for rainbow trout are residents. So rainbow trout do not go out to the ocean. They stay in the freshwater their whole time. That's another interesting area of interaction that we're not going to get to in this particular paper, but there's been a lot of research going on talking about what makes a steelhead, because a steelhead can have offspring that end up staying and becoming rainbow trout, or a rainbow trout can have offspring that end up migrating out to the ocean and become steelhead. So there are some genetic factors, but also some environmental factors that determine whether a rainbow trout ends up being anadromous and becomes a steelhead, or if it stays a resident. Another term that comes up in the introduction is the difference between emigration and emigration with an E. And we talk about this in terms of the spawning grounds. So if the spawning grounds are upstream here, when adults are returning to spawn, so they're going to the spawning grounds, they're immigrating with an I. When they are leaving the spawning grounds, it's emigration with an E. So when they're leaving the spawning grounds, they can either be talking about juveniles that are leaving to go out to the ocean, or adults. These would be kelts. So these are adults that already spawned in the spawning ground, survived, and then are emigrating out back to the ocean to grow and replenish, and then again returning as the next spawning season as an adult, immigrating back to the spawning grounds. Fecundity is another term that comes up. That's simply the number of eggs that a mature female has. And so if you have high fecundity, you have a lot of eggs, there's a lot of reproductive potential to produce a lot of offspring. If there's low fecundity, it means that you have fewer eggs, which means that you are limited in the capacity of producing fewer offspring. Anthropogenic is a fancy word for human-induced. So this is usually referred to for specifically environmental things. So if it's an anthropogenic impact, it means something that's caused by humans. So human anthropogenic climate change, for example, is human-induced climate change. Plastic in water bodies, that's anthropogenic. If you have a mudslide, if it's just a natural mudslide that's naturally occurring, that's not anthropogenic. But if it's a mudslide that was caused by this road being built here, or because we logged everything upstream, that would be an anthropogenic impact there. You will also see this denotation of n equals throughout the paper, and that's simply referring to sample size. So n equals 5 means that there were 5 individuals in that sample, whereas n equals 50 means that there were 50 individuals. So n equals 5 is a very small sample size, whereas n equals 50 is a larger sample size. And so the larger the sample size, the more trustworthy the research results is. If you're doing a sample or doing an experiment on 5 fish, that's not going to be as trustworthy as if you were doing an experiment that included 5,000 fish. Oops. And then lastly, there's a term that's going to come up as well called a pit tag. So a pit tag is this little tiny glass enclosed tag, so it's really small, that's inserted into a fish, and it's used to track the fish. We're actually going to learn how to pit tag next year in Aqua 250. Pit tags have individual numbers, so each pit tag is an individual number, and when they pass over a detector, that pings in the detector, and you can see that individual fish migration. So this is a really powerful tool in fisheries management, and it's actually exactly the same technology as microchipping your cat and dog. So same thing. So we're basically microchipping fish so that we can see individual migration pathways.